This is going to be a little recording here of the videos that are in Chapter 8 that we missed or were not in the posted PowerPoint lectures for Chapter 8. So I'm just going to proceed right through. So we're just going to look at the videos, if I could get to them. Here we go. So the first one is just showing cell division mitosis in fertilized sea urchin eggs, and there's no narration here, but just watch how the cells divide. And this is mitosis, cell division. See the cell undergoing there, cytokinesis, telophase cytokinesis, and then cell division, and boom, you get two daughter cells from the original parental cells. And then they divide themselves, and they get four. And this is further along down the developmental line, lots of cells here, and they're all going to be undergoing mitosis, cell division. So if you remember, mitosis is for organismal growth, tissue damage, repair, maintenance of the organism. Okay, so early embryological development in sea urchin through cell division called mitosis. You can see the early developmental stages here. And we can really just see a bunch of cells moving around and some cell division, especially earlier in this little video. Mitosis, cell division. Okay, where's our next little video? There's maybe 3 or 4 in this chapter. Here we go, nice narrated video on mitosis. Same type of cell division as we saw in that previous little video with the sea urchin. This one's narrated.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of 2 identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of 2 sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Okay, so again, little video there on mitosis. Here's a non-narrated video, also mitosis cell division. It's an actual living cell, and it's not colorized, but here in the central part is the nucleus, and you'll see the chromosomes condense and then organize themselves, condensing in prophase. Then in metaphase, they'll be in the middle. Anaphase, they move apart, and then telophase and cytokinesis is pretty quick as the cell divides in divides into two new daughter cells. And, well, let me get this to start here if I can for you. There we go. And then the cytoplasm of the cell is surrounding it. Notice the chromosomes, are condensing in prophase, organizing. You can't see the spindle fibers moving them about, but you can see them moving. They're getting organized to get into the middle, metaphase, coming up pretty quick here. Looks about almost there, metaphase. There we go. See how they're kind of lined up there in the middle? And then as soon as you start to pull apart, anaphase, A for apart. M, middle for metaphase. There's anaphase, see that? Moving apart, the chromosomes. Then notice cytokinesis, telophase, and then cytokinesis splitting the cell into two. And boom, just done. So two new daughter cells that are genetically identical to the original parental cells, ignoring any potential mutations. And that's just a little GIF showing, again, mitosis. And cytokinesis, which is the physical separation of the one original parental cell into two daughter cells. This is fairly short.
In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating two identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the two daughter cells. The cell plate's contents join the parental cell wall. The result is two daughter cells, each bounded by its own continuous plasma membrane and cell wall.
So notice a major difference between cell division and cytokinesis in animal versus plant cells. Plant cells must deal with the external, relatively thick and heavy cell wall, which would be a problem for traditional cytokinesis as we see in animal cells. So the solution for plant cells is they lay down cell wall material through vesicles in the middle of the cell, like the metaphase plate. That material builds together, forming a solid cell plate, and ultimately when it hits the cell wall, forming new cell wall material in the middle of the original one parental cell, and doing so cutting the cell in half, basically forming two daughter cells. And then new plasma membrane, cell membrane lines the inside of the new cell wall, forming two daughter cells. So one of the main differences in cell division between animal and plant cells. All right, we've got a couple more here, I believe. At least I think so. Meiosis. This one's a little bit longer, so we'll cover it in the next lecture part.